Welcome to Chapter 15 on the microbial mechanisms of pathogenicity. So what does that mean, pathogenicity? Well, this is the ability of a microbe to overcome the host defenses and cause disease. So the way I remember is this word is pathogenicity. It's the ability of the microbe to be a pathogen. And what is a pathogen? Well, we just learned a pathogen is anything, or is a microbe that causes disease. So the pathogenicity is the ability of a microbe to cause disease. And that's measured in virulence, and that's the degree of pathogenicity. And that's based on the presence of virulence factors. So different microbes will have the presence of different virulence factors, and virulence factors cause the microbe to have virulence or to increase its virulence and be pathogenic. So examples of virulence factors that microbes may have are all shown here. I'm just going to point out a few of them. So for example, the flagella. The flagella is going to increase the motility of a microbe. Well, by being more motile, it's going to be more able to invade and spread infection in the body. So by having the presence of the flagella or this virulence factor, it's causing it to be more virulent and more pathogenic. Another example is the presence of exotoxins. So these are toxins that the microbe may produce that are obviously toxic. We've talked about plasmids. These are a great example of a virulence factor. So the presence of a plasmid can increase the virulence of that microbe. They might have adhesion pili. That's going to cause that microbe to be able to adhere better. And if it can adhere to the host, it's more likely to cause infection. Presence of a capsule. Capsule is another example of a virulence factor. The capsule is going to cause it to be resistant to phagocytosis. So that's, of course, going to make it more virulent because it's harder for the immune system to attack it. So that's what we're going to be talking about. This whole lecture is talking about different ways or mechanisms that microbes can be more pathogenic. So here's this infectious disease process. So it's the process in which microbes cause disease. So first, they enter. So they can enter the body through these different ways. We're going to talk about those. Whether or not they are able to overcome the immune system and cause disease is based on different factors. That can be based on how they entered into the body. It can be based on the number of invading microbes. So the more microbes that there are that's invading the body, the higher the more likely they are to overcome the immune system, as well as the presence of other virulence factors, of course, is going to make them more likely to be able to evade the immune system. They're showing adherence here. Then, if they are able to overcome the immune system, they're able to evade the host defenses. These are different things that are going to help them do that. Then they're going to cause disease, and they're going to do that through damaging the host cells. These are different ways that they can damage the host cells. And then those microbes will exit, or they will try to infect someone else. And they're usually going to exit in the same way that they entered the body. So first, we're going to start with portals of entry. So microbes can enter the body through mucous membranes. This is one of the most common ways. So it can enter through the conjunctiva in the eye. The respiratory tract is very common. So we're a droplet inhalation or in airborne dust particles. An example of this is COVID. Can enter through the GI tract. So if we eat contaminated food or drink contaminated water, or just put our fingers in our mouth that are contaminated, that's going to lead to those microbes entering the GI tract. And lastly, we have the genitourinary tract. is another way that microbes can enter in the body. So those are all mucous membranes. Hopefully you remember that mucous membranes are membranes that are exposed to the outside of the body. So that's why it's such a great way for microbes to get inside the body. Another method is through the skin. So different infections of the skin, those microbes can enter through hair follicles, also the ducts of sweat glands, and some even grow in sebaceous glands. And the last route is the parenteral route. So this can be through wounds or trauma. So the microbes are going to get in through a wound, or it can be through an injection, or also through an insect bite. All right, so as I mentioned, whether or not a microbe overcomes the host defenses can be dependent upon the number of microbes that were invading the body to begin with. And then also, different species are going to have the ability to overcome the host defenses with different levels of initial infection. So some are better at that than others. And we use these two different measurements to measure those. So the first one we're going to look at is the infectious dose. So we call that infectious dose 50. And what it is the infectious dose that will cause disease in 50% of the hosts. The reason it's 50% of the hosts is like average. We can't have a constant number for all patients because different patients are going to have different immune levels, and different defenses. So, but in your average patient, the infectious dose 50 is going to cause disease in your average patient. So for example, down here, we're looking at Bacillus anthracis. So this is the microbe that it's actually a bacteria that causes anthrax. It has 3 different portals of entry, which is going to cause 3 different diseases. But we see also here that depending on the portal of entry, it has a different infectious dose. So we see that it's most effective in the skin. And so if it enters through the skin, it's going to cause these large black boils. And it only is going to take about 10 to 50 endospores to cause disease. And remember, disease means we're showing symptoms. Bacillus anthracis can also enter through inhalation. It's going to cause like flu-like symptoms, coughing. This is actually the most deadly form, but that requires 10,000 to 20,000 endospores to cause disease. So that's the initial infectious dose. So the patient would need to inhale that many endospores for the average patient to get sick. And then it's an even higher number if it's ingested. And if it's ingested, that causes diarrhea and digestive issues. Now, the other thing is the lethal dose. So this is the dose that's required that was going to cause death in 50% of the hosts. So infectious dose is the initial amount of microbes needed to cause death in your average patient. All right, looking at some more of these virulence factors. So this is adherence. If a microbe is able to adhere to its host, it's going to be more likely to cause infections. And so it can adhere by binding between these adhesions on the surface of the microbe. So we have adhesions on the surface of the microbe that's going to bind to receptors on the host cell. I think it shows it better on the next slide, so I'm going to go ahead and move on to that. So here, what we're looking at here on this picture, this is the cell membrane of the host cell. And here's receptors in the cell membrane, that's purple. And this is a microbe. And these are the adhesions in green. So the adhesions are molecules on the surface of the pathogen, and they will adhere or bind to receptors on the host cell membrane. The adhesions on the microbe surface can either be found in the glycocalyx or in the fimbriae. And they are going to bind to receptors, like I said, on the cell membrane of the host cell. Now, these receptors are receptors that are naturally always there. And the microbe has evolved to form adhesions that will bind to the receptors of the host cell membrane. Biofilms is another big one that aids in adherence. So remember we talked about biofilms before, and they provide attachment, and they also help to provide protection from antimicrobial agents. All right, so a couple of more factors. Let's see, go back, okay. So one is capsules. We've touched on capsules before. So capsules are going to help to evade host defenses. So it's going to stop the immune system from effectively being able to stop the microbe. So the capsule is going to protect against phagocytosis. Remember, phagocytosis is an immune cell that's going to come and engulf that microbe. But the capsule will block it from doing that. It's also going to block antimicrobial peptides, block complement and opsonization, and it's also going to block the inflammatory response. We're going to talk more about all these processes in the next couple of chapters. Another factor is the M protein. So the M protein is found on the surface of the microbe. More particularly, Streptococcus has an M protein, and it helps to resist phagocytosis, just like the capsule, and it also aids in adherence too. So it's like a double whammy there. All right, another way that microbes evade the immune system is through antigenic variation. So what that basically means, what's happening here, before we go into that, we need to know what an antigen is. So an antigen is anything that's going to cause an immune response. So it can be a protein, a carbohydrate, it can be anything. So what they're depicting here is a microbe, and it has these molecules on the surface. And these are called antigens because these are going to be recognized by the immune system as non-self, and then it will be attacked. So that's what happens. The immune system learns about these antigens here. It produces antibodies that will then bind to that antigen, mark it as non-self, and it will kill it. So the microbe responds by changing the antigen. It change the shape of its antigens on the surface. And so now the immune system can no longer recognize that antigen. And so it can go, the microbe can now go along unrecognized. Eventually, the immune system catches up, makes antibodies against that new antigen, and then it's going to be able to kill that microbe, which will then evolve again. So this is the process of antigenic variation. It's constantly changing and evolving its antigens so that it can evade the immune system. All right, a couple of ones that help to penetrate the host cell. So these are called invasins. A couple of examples, Salmonella and E. coli will alter the cytoskeleton protein. So if you remember from anatomy, probably you went over the cytoskeleton. It's made of actin. So the Salmonella and E. coli can actually change the actin to make it form this little pocket, or they call it a basket. And that's going to allow the microbe to like settle in. This is the cell membrane here. So it's going to settle in the plasma membrane and then eventually be able to enter the cell. Shigella and Listeria will actually use the actin as like a highway to move from one host cell to another. So they're going to take advantage of the cytoskeleton. All right. So now they've entered and they've invaded the host immune system. Here's a few ways that they can now harm the host. So they can harm the host by using the host's nutrients. So we're going to talk about iron. They can also directly damage the host cells and produce toxins. We're going to go through all of these. So like I said, they can use the nutrients, the host nutrients. So an example is Cyderophores. So one of these nutrients that's vital for both the host and for microbes is iron. So what Cyderophores do is they're produced by the microbe. They go out and bind to the iron, and then they bring it back across the cell membrane so that iron can then be used for microbial metabolism and growth. So it's going to take iron away from the host. Another thing that does is it removes the iron and doesn't let it be used by the resident microbes. So resident microbes want iron too, but if these pathogenic microbes are taking all the iron, they're no longer going to have iron. So then the resident microbes will die, which will give more nutrients and more space for the pathogenic microbe. All right, several enzymes that cause damage. You do need to know all these enzyme names and what they do. First, coagulase. So coagulase is going to cause blood clot formation. So what a microbe would want to do with that is it will release coagulase, which will cause a blood clot to form around it. Well, that's going to protect it. It's like making its own protective coating to protect it from other immune cells coming to attack it. But on the other hand, the body can also use blood clots to help to kind of isolate microbes. And so this is one of the host defenses, is it will use blood clots to isolate an infection in a certain area to keep it from spreading to the rest of the body. But the microbes will respond by releasing kinase, and the kinase is going to break down that blood clot that the host just made so that the microbe can then be released from that area and spread to the rest of the body. Hyaluronidase is a spreading factor. So if you remember that connective tissue has hyaluronic acid in it. Microbes that are wanting to evade through the epithelial cell layer, and they're trying to get through the connective tissue, they will produce hyaluronidase, and that will break down the connective tissue so that they can then spread through the connective tissue and penetrate and spread into the rest of the body. Collagenase is pretty much the same thing. If you remember that there is the protein collagen in connective tissue as well. So collagenase is going to do the same thing, but now it's breaking down collagen to break down the connective tissue so that it can spread to the rest of the body. And lastly, IgA protease. So IgA is an antibody that the body produces, particularly in mucous membranes, which we know mucous membranes is an area where microbes are entering most commonly through. And so what those microbes will do in that area is they'll produce IgA protease, which will break down those antibodies. Without those antibodies, then the immune system will not be alerted to the invasion of a microbe. And so then the microbe can go undetected. So here are some illustrations of those enzymes. So coagulase, remember, is producing blood clots. So here we have pathogens that are releasing coagulase, and that's going to cause a blood clot to form around it. And that's going to protect it from phagocytosis and other immune responses. The kinases, this is a more specific example of a streptokinase, but that's an example of a kinase. It's going to dissolve a blood clot that perhaps the body produced. So here we see the pathogens out here in this area not yet in this blood vessel, because the body had produced a blood clot around the pathogens to try to isolate it in that area. But the microbes produced the kinase, which broke down the blood clot, which then allowed them to spread. But now we're looking at the use of hyaluronidase. So here is our epithelial layer, connective tissue down beneath. The microbe will produce hyaluronidase, which will break down the connective tissue so that it can then spread through. All right, moving on to toxins. So toxins are poisonous substances that are produced by microbes. They can produce different symptoms depending on the toxin. So they can produce fever, cardiovascular problems, diarrhea, and shock. These are just some examples. A few definitions before we move on. Pathogenicity is the ability of the microbe to produce toxin. Toxemia is the presence of toxin in the host's blood. And intoxication is the presence of a toxin without microbial growth. So this can happen if you have microbes that produce a toxin, say in your food. And then you cook the food, so you kill all the microbes. So when you eat the food, you're not getting infected with microbes, but those toxins are still in the food. So then when you eat the food, those toxins weren't neutralized. And so then you still have the symptoms of like if you had the infection, because of those toxins are still active. There's two types of toxins. There's exotoxins and endotoxins. Exotoxins, think of as like outside of the microbe, and endotoxins are more like inside. So that might help you keep those two straight in your mind. Exotoxins are intentionally produced by the microbe to be secreted outside of it. So the microbes are producing these toxins, these exotoxins, to be secreted as part of their normal metabolism, so that those exotoxins will then harm the host by destroying the host cells and inhibiting metabolic function. Both gram-positive and gram-negative bacteria can produce exotoxins, as well as other types of microbes, like funguses can produce toxins or exotoxins as well. And since the exotoxins are outside of the cell, they can be neutralized by antitoxins. So these are antibodies against those toxins that will neutralize them and neutralize their effects. On the other hand, we have endotoxins. So if you remember when we were talking about gram-positive versus gram-negative, we talked about how gram-negative cells have the thin peptidoglycan layer, and they have the outer membrane. And how the outer membrane had the LPS here, that had the lipid A down in the bottom. The lipid A, we mentioned how the lipid A produced or was an endotoxin. Now this lipid A here, it's part of the cell. It's part of that microbe. It's not produced to be released, it's produced and made to be part of the microbe. So the only time that the lipid A is going to be released and cause toxic effects is if that cell dies. And when it dies, then it's going to break apart and it's going to release those lipid A's. We're only going to find endotoxins in gram-negative bacteria, because only the gram-negative bacteria have the outer membrane with the lipid A. What we see with endotoxins is that the lipid A is going to cause a widespread inflammatory response, and it's actually this inflammatory response that leads to so many issues such as shock and death. All right, so let's talk a little bit about exotoxins. So different examples include genotoxins. So this type of toxin is going to damage the host DNA. There's also membrane disrupting toxins. So this type of toxin is going to lyse the host cell by disrupting the plasma membrane. An example of this is hemolysins, which is going to lyse erythrocytes or red blood cells specifically. And we are going to see hemolysins in the lab. Super antigens, these are going to cause an intense immune response. So it's the immune response that's causing an issue with the super antigens. This causes fever, nausea, vomiting, diarrhea, shock, and death. So this is just a review of all of the things for exotoxins. So they're produced by both gram-positive and gram-negative bacteria and fungi and protozoa. They're a byproduct of a metabolism. So they are produced to be secreted to have their effects on the host. And they're proteins that are being produced and secreted. They don't cause fever or the immune response. They can be neutralized by antitoxin, and they have a low lethal dose, so they're much more potent. And they have different effects. So there's neurotoxins, so toxins that are going to have an effect on the nervous system. Enterotoxins, hepatotoxins are going to have an effect on the liver. So different microbes are going to produce different toxins that have different effects. A few examples. So the botulinum toxin is a neurotoxin. And we also see different infectious doses for these different toxins. So that has a very low infectious dose. It's actually one of the most potent toxins that there is. The Shiga toxin is an enterotoxin. So enterotoxin is going to have an effect on the GI tract. So this is going to cause diarrhea. Can also cause a bloody diarrhea and pus in the diarrhea. And we also have staphylococcal enterotoxin. So that's another enterotoxin that causes diarrhea. So looking at the infectious doses, these would be getting more and more potent, or less and less potent as we go down, because this one requires a lot more of that toxin to cause disease. So here's that cholera toxin. So it's caused by a Vibrio cholerae. That's the microbe that's producing the cholera toxin. The cholera toxin will bind to a receptor on the cell membrane. That's going to cause the release of all these ions in the epithelial cells in the gut. And then water is going to follow those ions. And then that leads to massive amounts of diarrhea and dehydration. And most commonly death if it's not treated. So endotoxins, remember those are the lipid A in our gram-negative bacteria. That's only going to cause disease if they are forced to be released through cell death. So that's going to be either, it could be cell death just from the immune system killing those cells, but we see a lot of it when they're killed very rapidly through antibiotics or if antibodies are used. So what happens, so this example here is a immune cell that's killing this cell, or killing the bacteria. And then it's going to break down the cell that's going to release the lipid A. The lipid A causes the cell to produce interleukin 1. Interleukin 1 is released from the immune cell, travels through the blood to the hypothalamus, and it stimulates the hypothalamus to produce prostaglandins. And prostaglandins is what leads to fever. So here's the summary for those endotoxins. Only going to be found in gram-negative bacteria because only the gram-negative bacteria have that outer membrane with the lipid A. So it's the lipid A that's causing it. It's part of the LPS in that outer membrane. It's going to cause fever. So that's actually what the main issue is the fever. They can't be neutralized by antitoxin, but they do have a high lethal dose. So it's going to require a large amount of that microbe, a high infection to lead to that. And also, it's most commonly where we have issues is if they are killed at a high rate. So if antibiotics are prescribed, that's going to kill those bacteria very quickly. They're all going to release their lipid-A's at the same time. It's going to cause a very high fever. All right, lastly, we're going to look at some of the pathogenic properties of different types of microbes. So we're going to start by looking at viruses. So viruses evade the immune system mostly because they're intracellular. We know that viruses have to replicate intracellularly because they cannot replicate on their own. And as such, then they're somewhat hidden from the immune response and from immune cells when they're inside of a cell, of the host cell. And so what they try to do, viruses have evolved to try to mimic and be the most like the host cell. So they're going to use the host cell enzymes. They're going to use the host cell ribosomes. They're going to bind to normal host cell receptors and just use all the host cell machinery. Some visible effects of a viral infection is called cytopathic effects. So what we're seeing here, this is what normal cells would look like. This is what cells look like with a viral infection. So a lot of cells, or a lot of viruses, when they infect the cell, when they're ready to, when they're done with the replication cycle, they'll rupture the host cell. So that's going to lyse and kill the host cell, of course. And so that's what we're seeing here, is we're seeing this clearance where these host cells have died because the virus has used that host cell to replicate and then lyse that cell to be released. And so that's what we call cytocidal. So it's killing those cells. And that's what we're seeing here. One thing that the host does in response to a viral infection is produce interferons. So interferons, so a cell recognizes that it is infected. It's too late for that cell, but it will produce interferons, which will go to another cell, a neighboring cell, and warn it. So if it gets interferons from its neighboring cell, then it will know that, look, there's a viral infection going around. Let's put up our defenses now before we get infected, basically. Pathogenic properties of fungi. So some of the products of fungal metabolism might be toxic. Also, chronic infections can cause allergic responses. Fungi can produce toxins, exotoxins that we just talked about. So there's examples of neurotoxins and hepatotoxins that are produced by fungi. Fungi can produce protease enzymes that help to facilitate adherence. And some fungi even have capsules, which is going to help it to prevent phagocytosis. Yeast infections. So these are an example of a infection by a fungi. We mostly see yeast infections in immunocompromised patients. It's a very common issue with immunocompromised patients. They can cause respiratory illnesses, meningitis, vaginal yeast infections, thrush in the mouth. These are examples of yeast infections. And then there's also protozoa and helminths. So these both are going to use the host tissue for growth. So it's going to use the host tissue for nutrients. And then their presence is going to cause an immune reaction. So the body wants to try to kill the protozoa or the helminths that are in the body. So they're going to mount an immune response against that. The metabolic products of these microbes can cause symptoms. They can avoid or evade the host defenses by some, especially protozoa, may reproduce inside of the phagocyte. So they'll be engulfed by the phagocyte, and then they'll just take over the phagocyte. So they don't say that's a problem. They're like, okay, fine, engulf me, but then I'm going to use you to divide inside of. They also can rapidly change antigen. So that's going to evade the host defenses. And they can also interfere with the host functions. So tapeworms is a worm that lives inside the gut, and it's going to use up all the nutrients of the host so that the host doesn't get any nutrients, so they're malnourished. Heartworm can block the arteries. Malaria replicates inside of red blood cells and then will burst them. And then lastly, after all of this, then the microbe is going to exit and then go to and infect the new host. It's usually going to exit through the same portal that it entered through. So if a microbe entered through the respiratory tract, then it's also going to exit that way through coughing and sneezing. Same thing with the gastrointestinal tract. So if it entered that way, then it's going to exit that way, either through feces or saliva. Urogenital tract is going to exit through urine and vaginal secretions. The skin can exit through contact, and blood can exit through insect bites, through direct contact with the wound, or through needles and syringes. And that's it. So those are the different mechanisms of pathogenicity. Hope you guys have enjoyed, and I'll see you guys for the next chapter soon.